Since 1928, antibiotics have saved hundreds of millions of lives, but bacteria are fighting back. Drug-resistant infections now kill over 1.2 million people a year, more than HY5 and malaria combined, and by 2050 that could reach 10 million. Scientists are reviving a nearly forgotten weapon, a virus that hunts and kills bacteria with surgical precision. To understand why this breakthrough matters, you need to see what we are up against. We do not simply kill bacteria, we drive natural selection at hyperspeed. Inside any bacterial population, genetic mutations occur constantly. Most do nothing, but occasionally, a single mutation protects a bacterium from a specific drug. When an antibiotic sweeps through, it wipes out the vulnerable population, leaving only these resistant mutants. With their competitors gone, these survivors multiply rapidly, passing on their defensive traits. We accelerate this evolutionary pressure every time antibiotics are overprescribed in clinics or used unnecessarily for viral infections. But the problem expands far beyond healthcare systems. Worldwide, over 70% of medically important antibiotics are used in agriculture, not to treat sick people, but to promote growth and prevent disease in healthy livestock. This constant low-dose exposure turns industrial farms into massive evolutionary laboratories. From there, resistant strains enter our food supply, soil, and water systems, spreading globally. What was once a localized defense mechanism becomes a widespread systemic threat, rendering our most powerful medicines useless against common infections. In hospital wards, this evolutionary speed translates directly to human suffering. Patients battling methicillin-resistant Staphylococcus aureus, or MRSA, face severe skin and bloodstream infections that bypass standard therapies, making it a leading cause of hospital-acquired sepsis. When carbapenem-resistant Klebsiella pneumonia infects patients in an intensive care unit, the mortality rate for bloodstream infections can exceed 50%. Meanwhile, Escherichia coli, carrying the NDM1 gene, destroys the effectiveness of almost all classic penicillin-like drugs, leaving clinicians with no viable treatment options for basic urinary tract or abdominal infections. Developing new compounds to replace these failing treatments is an economic dead end. Bringing a single novel antibiotic from laboratory discovery to market requires more than 10 years and billions of dollars in research and development. Yet, because bacteria replicate so quickly, resistant strains often emerge within just a few years of a new drug's launch. Pharmaceutical companies cannot recover their massive initial investments on a drug designed for short-term use that targeted microbes will quickly bypass. Simply manufacturing more drugs cannot solve a crisis driven by the fundamental mechanics of evolution. In 1915, English bacteriologist Frederick Twort observed something strange in his laboratory dishes: glassy, clear areas where bacterial colonies had vanished. 2 years later, French-Canadian microbiologist Félix Terrel made a similar observation while studying dysentery patients in Paris. He recognized that an invisible microscopic agent was destroying the bacteria, and he named this entity a bacteriophage, literally, a bacteria eater. These agents are highly specialized viruses. For medical therapy, scientists rely on lytic phages rather than lysogenic ones. The distinction is functional. Instead of integrating quietly into the host's DNA, a lytic phage acts as an active predator. It lands on a bacterium, binds to specific surface receptors with high precision, and injects its genetic material. Once inside, the phage hijacks the cell's metabolic machinery to manufacture hundreds of copies of itself. The cycle ends when the phage produces specialized enzymes that puncture the bacterial cell wall, causing the cell to burst open and release the new viral offspring to infect neighboring targets. This process is highly selective. Because phages require specific receptors to bind, they target only the problematic bacterial strain, leaving human cells and the surrounding protective microbiome untouched. But when cheap, easily standardized penicillin arrived in the 1940s, Western medicine abandoned this complex, strain-specific therapy. In contrast, researchers in the Soviet Union and Eastern Europe continued to develop phage therapy, keeping the science alive at centers like the Eliava Institute in Georgia, while the West relied almost entirely on chemical antibiotics. The limitation that once marginalized phage therapy in the West, its extreme strain-level specificity, is now being solved through a modular, modern toolkit. Rather than relying on a single highly specialized viral strain, researchers now design precision phage cocktails. By mixing multiple distinct phages into a single therapeutic dose, clinicians can broaden the target range to cover multiple bacterial variants, while simultaneously preventing the pathogens from evolving resistance. If a bacterium mutates to escape one phage, the others in the cocktail are there to eliminate it. Apart from natural cocktails, genetic engineering has turned phages into programmable weapons. Using CRISPR-Cas systems, scientists can now modify phage genomes to slice through essential bacterial survival genes, or even specifically target and destroy the plasmids carrying antibiotic resistance genes. This engineering prevents the bacteria from deploying their standard defense mechanisms. These phages do not have to work in isolation. When combined with traditional antibiotics, they often exhibit a powerful cooperative effect known as phage-antibiotic synergy. In this dual attack scenario, sublethal doses of antibiotics can stress the bacteria, causing them to swell and accelerate phage replication. This combined force is particularly effective at dismantling biofilms, the slimy protective shields that bacteria construct to protect themselves from drugs. Phages produce specialized enzymes that chew through this protective matrix, exposing the vulnerable bacteria underneath. With over 30 clinical trials active worldwide, an April 2026 Medscape report noted that clinical momentum is shifting rapidly, turning a century-old biological anomaly into a scalable, high-tech weapon against the superbug crisis. Locus Biosciences is advancing LBPEC01, a CRISPR-enhanced phage cocktail, through Phase 2 and 3 clinical trials. Under the Eliminate program, this engineered therapeutic targets antibiotic-resistant Escherichia coli, strains causing chronic urinary tract infections. Meanwhile, in Germany, researchers with the Phage for Cure initiative are developing inhaled phage cocktails specifically formulated to clear persistent Pseudomonas aeruginosa lung infections in patients with cystic fibrosis. These structured trials run parallel to expanding real-world observational data. A multi-country retrospective dataset tracked 100 personalized phage treatments administered across 12 nations, reporting clinical improvement in 77% of cases and successful pathogen eradication in 61%. This growing clinical evidence is backed by powerful, compassionate use recoveries. In the United States, a teenage girl suffering from a highly resistant, life-threatening infection became a landmark case at UC San Diego. After receiving emergency intravenous phage therapy, she achieved complete recovery, a success that galvanized modern clinical interest across the country. While these individual outcomes and early trials show immense promise, they also serve as a reminder that the broader evidence base is still developing. Deploying this biological precision in clinical settings is technically demanding. Because a single phage targets a highly specific bacterial strain, doctors must isolate the patient's exact pathogen and screen it against viral libraries to find a match. This matching process requires specialized laboratory equipment and valuable time that critically ill patients often lack. Currently, regulatory pathways in the United States and Europe are still developing, leaving these therapies experimental and difficult to manufacture at scale. Because these treatments are not widely available, this documentary is purely informational, does not constitute medical advice, and patients must always consult licensed healthcare professionals. Major medical centers are organizing dedicated phage banks to cut matching times from weeks to hours. Alongside these banks, researchers are building engineered phage libraries to target the most common resistant strains quickly. Regulators in both the United States and Europe are slowly developing fast-track approval pathways for these adaptive biologics. The most realistic path forward is not the total replacement of antibiotics, but collaboration. In the near term, antibiotics and phages will likely share the front line of infectious disease care, combining their strengths to defeat superbugs that neither can destroy alone. As superbugs outpace our drugs, medicine's next chapter is not about inventing stronger synthetic shields, but collaborating with nature's own precision predators. In this microscopic arms race, our future lies in partnering with our oldest allies. Subscribe for more breakthrough science.